How's that?
Oh, that's better.
Good.
Yep.
Kia ora. Sorry, everybody. We're off. Technology. My apologies. So just a brief starting point. So there's just a few slides around some of the major issues. Okay, so the first point I think is worth us always making is that the fact that we've actually got COVID vaccines within a year is an incredible scientific feat. And that I've got a slide later on to explain why we've managed to do it so fast. But at this point, there is a huge amount of vaccine types available. And on the right hand, there are all the different vaccine types available. And when this one was drawn up, there were actually 20 in the large phase 3 trials, which is absolutely unheard of to have so many going through phase 3 trials. Normally, a vaccine in development has one large phase 3 trial. So it just shows you the amount of international resources that was thrown at developing COVID vaccines. And the different types are listed on the right, and we'll focus on the RNA vaccines. And the other key ones at the moment are the non-replicating vector vaccines, which many of you will be aware of, the Janssen and the AstraZeneca. And then coming along after that are the inactivated vaccines and the protein subunits. The protein subunit is the Novavax, which is the other one New Zealand's got an advanced purchase agreement with. These others are not likely to come so soon. And the key point about the development of vaccines for the COVID virus is that this is a little schema of the virus. And you can see this little outer spike protein. Now, a lot of you would have heard a lot of conversation about the spike protein. That's the part of the virus that is identified as the antigen, as the foreign material. So that's the part of the virus that all the vaccines are directed at designing to copy some aspect of the spike protein to get the body immune response to it. So they're all focused on the spike protein. Now, the vaccines we're particularly interested in New Zealand, and the one we're privileged to have right now, what we call nucleic acid vaccines. Now, the advantage of these vaccines is that they don't have any virus in them at all. They're not live, they're not even subunit. They're actually coding for the nucleic acid bit that codes for the spike protein. So the pros are a huge advantage. There's no risk of vaccine causing disease. The other pro is because it's the genetic code that you're making, they're actually very quick to design and manufacture. And if we did get significant mutations in the COVID virus, they're also very quick to replicate with a mutation, which is likely to be a big advantage. The disadvantage is that RNA vaccines are new and that they have been in design for a long time, but they're relatively new as a human vaccine. And of course, the big disadvantage for New Zealand is the cold chain issue, that RNA is a relatively unstable beast, and that's why it's lipid coat to stabilize it. But they do require very careful vaccine storage, which makes a difference to distribution. And that really is quite a challenge for New Zealand. Now, this is a little schema to explain how an RNA vaccine works. Now, the RNA vaccines are ahead of the market. I think it's very unlikely we will see DNA vaccines. So how does an RNA vaccine work? Well, you can see from this little schema that you actually code the genetic RNA piece for the spike protein. Now, RNA has been around for a long time as a concept with vaccines, but the big science breakthrough was it's encased in a lipid coat, in a fat coat. Now that stabilizes it so that when you inject the RNA, it hangs around for long enough for the body to create proteins from and then create the immune response. So essentially you coat your RNA in a more stabilizing lipid coat, you inject it, it's taken up by the body who then creates the viral proteins, the spike proteins. So the body itself is generating spike proteins, and then the body responds, the immune system, like that's a dendritic cell, to the spike protein. So the little summary is you've got a lipid bundle containing your fragment of RNA, which gives instructions to the cells to produce the protein. So very, very beautiful technology. Now, the importance of this being RNA, messenger RNA, for those of us who remember our basic school science, RNA does not go into the cell nucleus. So there is no ability for these cells to get in and alter the human genome in any way. It's a really important point, and a point that I think is useful to make in all our teaching so that we don't then get the myths created around whether it can genetically affect us. I think we're all aware that New Zealand has got advanced purchase agreements with the Pfizer BioNTech, which is the RNA vaccine, with 2 vector vaccines, and with a protein subunit vaccine. So that's just to show you, those are the ones that New Zealand could potentially purchase going forward. But at this point in time, the only vaccine we've purchased is a messenger RNA COVID vaccine. That's the way it's described in the handbook. The company calls it Community. That seems to cover RNA immunity and community. I believe that's how they came up with the name. And it's publicly known as the Pfizer BioNTech vaccine. 2-dose schedule, a minimum of 3 weeks apart. The point about the minimum of 3 weeks apart is there's no problem in giving the second dose at a longer gap. There is a problem in giving the second dose closer than 3 weeks. So the minimum of 21 days, no problem if you have to delay it. Particularly in New Zealand environment where there's not an urgency, where people are not being exposed, you were better off having a delay rather than giving a vaccine early. Multi-dose 5, dose of 0.3 mils. So people often want to know what are the active ingredients in Community. So because it's a very beautifully designed RNA vaccine, there's actually not a lot in it. So you've got the fat coat on the outside, and there's not a lot in that. There is a product in this called Polyethylene Glycol. This is very commonly used in many, many products in the world, commercially and in medical products. Very rarely, there's probably a handful of people in New Zealand who have anaphylaxis to polyethylene glycol. So it's worth being aware of that. The rest of these are in your human body anyway. The salts that keep it at the same pH as the human body pH are salts that are within our body anyway, so would be no problem. And the sucrose, which is also a common element in the human body, is to help it stabilize at the ultra cold temperatures. That's essentially what's in the vaccine. So it's a nice, simple vaccine with not a lot of ingredients. Now, the key big questions. First one is, does this vaccine work? Okay. Well, firstly, all vaccines, as you're all aware, are only gone through to licensure after they've done animal studies, phase 1, phase 2, and large phase 3 clinical trials. So the large phase 3 clinical trials for this vaccine were undertaken last year. They started in July 2020. And the reason they were finished so quickly is essentially because there's so much COVID out there and a lot of people were willing to be recruited. So they got to their endpoints very, very fast. So by the end of the year, vaccine efficacy, as we call it in clinical trials, where you give randomly half of the group the vaccine and half of the group a placebo. In this case, it was saline. They were done in 6 international sites all around the world with diverse ethnic cultural groups. Obviously, there were not Māori included because these are big international countries outside of New Zealand. New Zealand couldn't run these studies because we don't have COVID disease. However, what it did show from this study, highly effective vaccine. 7 days after the second dose, you had an estimate of somewhere between 90 to 97% effective against symptomatic disease. So incredibly effective vaccine. It was also shown to be effective in the elderly and across people with medical problems. The study was not done in children under 16 because the urgency of the first clinical trials were to protect those at highest risk. Those at highest risk of COVID are people with severe disease, elderly, people with medical problems, not children. So the original clinical trials were done on adults. Now, I can report today, and I don't have it in here, that the clinical trials on children aged 12 to 15 have just gone through, and it's looking really positive for 12 to 15. And they are currently recruiting for clinical trials on children aged 6 months to 11. So we're hoping within a few months, we should have clinical data on children, which would then have to go to MedSafe for proper tight scientific scrutiny before New Zealand makes a decision on whether we can also use these vaccines in children. So clinical trials, highly effective. But even more than that, the advantage of New Zealand not having to rush ahead and be the first country in the world to use these vaccines is that we were also then able to look at the international data on countries that have been using this vaccine. So this vaccine's now been out there in the world since December. I understand up to recently more than 150 million doses. The need is so great that once these countries got licensure, they rocked out with this vaccine very fast. We now have really strong clinical data from quite a few countries looking very closely at the effectiveness. So this is published data from Israel, which is probably the country in the world to get the highest coverage very fast, showing that in real life, the documented experience is the same as the clinical trial, that you're still seeing well over 90% effectiveness against symptomatic disease in all age groups, including elderly people and across people with medical problems. When they looked across their data, if people had 3 or more medical problems, possibly the effectiveness was slightly less, but still highly effective. The effectiveness also, you get reasonable effectiveness even after the first dose. And they did have data in this, plus other data we've got from America and from Scotland, showing this vaccine also does reduce your chance of carrying it and spreading it to others. Now, it doesn't totally stop it, but it does reduce it. So this vaccine clearly works highly effectively against symptomatic disease, and it has some degree of effectiveness against asymptomatic. So does it work? Not only does it work, but it works incredibly well. So this is way more effective than most vaccines we're currently using. It's up there with the effectiveness, probably the MMR vaccine, way more effective than many of our traditional vaccines. So highly effective vaccine. Next big question, how can I be sure it's safe? Well, once again, you've got the clinical trial data. And so I've listed a bit of the clinical trial data here. Clinical trial data was very comforting, showing that it looked very safe in the original clinical trials. Following on from the clinical trials, this vaccine has been used so widely that countries with resources are looking very closely at its safety profile. And since, we've been hearing about the AstraZeneca story lately, the safety profile of these vaccines are being scrutinized. Now, I present at the bottom of this study, at bottom of this slide, some US data just out of interest. So where they looked closely at more than 12 million doses. Now, these are eye-watering amounts for vaccinology. This is huge amounts of safety data. I can't say strongly enough that we have got very, very large amounts of safety data. It's not just passive, it's also active safety data. So from the American data, the incidence of anaphylaxis is around 4.7 per million. So anaphylaxis rates are slightly higher than childhood vaccines and flu vaccines, but they're well within the range of what you'd expect with a biological product. So we will see anaphylaxis and we need to be ready for it. Other than anaphylaxis, across all this data, there were no disproportionate events and conditions of interest. Now, the Americans have been looking in a whole lot of different ways, and I just note one comment at the bottom. They did do a sub-study on large amounts of very frail elderly, because in the early days, there was concern about whether very frail elderly were more likely to die after the vaccination. Very reassuring data, following up actively, that there's no evidence of an increased incidence of death for 30 days after a vaccine in very frail elderly. Now, I just reflect some of the American data here. You can see that they've got very large numbers. The Mrna is the other RNA vaccine used in the States. So these are very large data that the Americans are actively following. The question that comes up all the time to us is, well, how do you know it will be safe in 30 years' time? It's all very well to say it's safe now, but how can you be sure? Oh, I've jumped to the different slide for that one. I'll come back to that one. How can you be sure this is going to be safe in 10, 20, 30 years' time? So what I would say from a biological plausibility point of view is that you would expect to see, with the very large numbers vaccinated, some difference in background rates of really important conditions: neurological, immunological, autoimmune mediated, Bell's palsy, multiple sclerosis, Guillain-Barré. So essentially the Americans and several other countries with resources, the UK, the Europeans, the Australians, many other countries, are looking at background rates of these really important conditions. Now, this is the sort of database they're using to scrutinize the AstraZeneca vaccine. So you can see the list of background conditions they're scrutinizing closely. Now, when you're talking at multi-millions of doses being given, you would have a warning signal if there's going to be an autoimmune or a neurological surprise from this vaccine. And so far, after these many multi-millions, there is no sign of any surprise. So we can say confidently that I'm probably looking at the best safety data I've seen previously from any vaccine that's got through clinical trials. This is not an experimental vaccine. This is no longer a brand new vaccine. This is a vaccine that is out there, used extensively in the world in doses like we've never seen before in a vaccine, closely scrutinized. So we can be very reassured about the clinical data safety of the important big issues. Now, next moving on is, well, what does it cause? Now, it is a reactogenic vaccine. This is a vaccine that does give us reactions post-vaccination event. Now, I've had my own first dose, and I know you certainly can get quite a deep ache and a sore arm. You can get a lot of common events like fatigue, headaches, myalgias, chills, fevers. I think it's important. This is the American data again. So this is on very big numbers of more than 12 million. You can see that, you quite commonly get these sort of side effects 1 to 2 days after the vaccine. And I particularly note, and many people have been reporting this locally, that it's more common with the second dose than the first. So I think we need to be reflecting that to people, that you do see reactions after this vaccine. It does show the fact that, immunologically, this vaccine is working very well, and you do see more doses, more reactions after the second dose. So I think we need to highlight that. Maternal vaccination, I just want to reflect that the original clinical trials did not include pregnant or lactating women. I think that's very frustrating because there's no biological plausibility concerns of giving a vaccine of this nature to a pregnant woman. You are not giving any part of the virus to the woman. You're giving a piece of genomic piece that then she creates her own proteins. And actually there's an advantage to vaccinating a pregnant woman because we know that the antibodies can cross the placenta and protect the infant. So since the clinical trials, it has actually been used quite widely in pregnant women because countries with a high risk of COVID, pregnant women are at high risk of COVID. So it has been used quite widely internationally now. And in the States, they now have a database of over 30,000 self-reported pregnant women that they've followed up, of which is over 16,000 are the Pfizer vaccine. And the reactogenicity adverse event profile is no different for pregnant women, and there's no different in the outcome rates in vaccinated pregnant women. So this is the current New Zealand advice about using this vaccination. Who should not have this vaccination? Anybody who should not have it who's had a history of anaphylaxis to any component or a previous dose of this vaccine. Now that's an incredibly small group of people. We haven't actually had any anaphylaxis cases yet directly to this vaccine. We've had one that was anaphylaxis type, but didn't quite reach the definition. So anyone with that history, we would not vaccinate, and we'd consider referral to an allergy specialist. So there may be the occasional very rare people out there who have anaphylaxis to PEG. Precautions is pregnancy because it wasn't included in the original clinical trials. However, as I've said, there's very reassuring clinical data out there now. So we are advising pregnant women that if you think you are at any risk of COVID, then go ahead and vaccinate. But if you're at no risk of COVID, then it's perfectly reasonable to wait till after you've had the baby. So that's a precaution. The only other precaution we've got is anyone, when we looked at the American data on anaphylaxis to this vaccine, about a third of the cohort had anaphylaxis to any previous biological product at all. So if anyone's had a history of anaphylaxis to any product, then certainly go ahead and vaccinate. It's not a contraindication, but we recommend close observation for up to 30 minutes, just in view of the fact that they have got a slightly increased risk of anaphylaxis over the general population. The people we would support proceeding with vaccination, as we've said, are pregnant women at risk of exposure. And the same as with flu vaccination, people who are on antithrombotics, people who have coagulation disorders or on blood thinners, we should proceed as we do with flu vaccination right now. So a lot of people should not get turned away. We've had stories of people getting turned away who we can comfortably and confidently vaccinate. I don't think I have it here, but we are strongly recommending vaccinating for women who are breastfeeding. There is no concern for a breastfeeder in receiving this vaccine. So I think you're all aware of the New Zealand vaccination strategy. I think we've all seen this. I've just put it in here just so we're all aware of it. But I think I'm talking to people who are very aware of it at the moment. I just wanted to also add a slide from myself about challenges, is that the world strategy for vaccination is essentially to stop people dying and get very sick. So New Zealand's in a very different position, that we are still trying to stop the spread of COVID in our community. So our strategy initially was to protect those who are at risk of being exposed to COVID, very different from many other countries. And of course, going forward, New Zealand has to decide, are we going to maintain an elimination strategy? Do we have the ability to do that? Can we stop COVID? Once we open our borders, COVID disease will flood back in. If we only open our borders to vaccinated, yes, the rates would be much reduced, but we will still see COVID in New Zealand. So what we can say at the moment is vaccines have really high individual protection, particularly against severe disease, and they do have a reduction in asymptomatic spread, but it will not be complete. We still don't have any data on children. The data on pregnant women is accumulating. We don't know if and when we will have to boost. We expect that we will have to boost at some stage, but we're just going to have to keep watching as to see when immunity starts to wane, when we will need to boost. We don't know differences between different vaccines. I think we're really, really fortunate to have a vaccine that's got a huge amount of clinical data behind it, and it's looking like one of the best performing vaccines in the world. So we can really celebrate that, but we don't know about different vaccines. I think we know a lot about logistical challenges out there, so I'm not going to. Now, common concerns, I've just slipped in a couple of slides about this. I'm going to show you a slide about people thinking that the development time's been too short. Special groups, elderly, pregnancy, children, immunocompromised. My point about immunocompromised is they're the people who are at highest risk of COVID disease. And so absolutely essential that they do receive this vaccine. There's no concerns about vaccinating immunocompromised, but clearly somebody who's severely immunocompromised may not get a good response to the vaccine. So you also want to ring protect them by vaccinating those around them. So this is the one slide about people say, well, this vaccine's been developed faster than usual vaccines. Well, it has, but the standard steps within the process of developing this vaccine have all been in place. So all the animal studies, phase 1, 2, 3, the large pivotal trials have all been undertaken. And the reason that we were able to get a vaccine way faster than we've ever done before is firstly, huge incentive. The world needed to solve their problem. We're in the middle of a major pandemic. People are dying. Huge amounts of resources were thrown at it. Science came together internationally in collaboration in ways that we've never seen before. So a lot of steps that traditionally you do one after another, after another, after another, they were able to run in parallel. And one example is the really big manufacturing plants that cost a fortune to make. They built these plants even before they knew if the vaccines were going to work. So that's a real example. So once the vaccine came off its clinical trials, they were able to immediately run through the scale up to manufacturing. Clinical approval processes were speeded up by, as the science was coming, they were feeding them straight through to the approval process bodies who could review it as it came through, rather than waiting till the end and putting the whole dossier together. So that's just a little picture of why, doing the same approach to vaccinology that we always do, we were able to get a vaccine in less than a year. Now, I don't want to spend a lot of time on vaccine hesitancy. I'd be happy to have a conversation on that at any stage. But I think one of the problems is the word hesitancy, that internationally we'd quite like to move away from it. But the more you keep saying who's hesitant, the more we create hesitancy as an issue. So this is a picture very recently out of the World Health Organization, looking at all the different key domains that make somebody less likely to accept a vaccine. So obviously within there, you've got how people think and feel, alongside all the social processes that affect how people think and feel. So if we just go out there and poll what people think and feel, but we don't add in the social processes, the motivation, the practical issues, then we keep coming up with these magical figures that, da percentage of our population is hesitant, that we have to focus on all these issues in the chain so that somebody is really receptive and able to receive a vaccine. And a lot of this is about our system. So the way we communicate also, I've just put that slide in here, but I think I've talked for long enough. So we are just starting to frame more and more messages about the way we communicate. So yes, how do we sell the science so that somebody feels both that they trust the science and that they feel confident and comfortable to ask questions and then to proceed. So a lot of what we're talking about now is the way we frame things. And so a lot of the conversation is around whether we can frame selling the cake for a value proposition for us as an individual and us as a community, and then embed within that these key important bits of science so that people feel that there is, you know, a value proposition to why they'd vaccinate. And then behind it all, there's trusting science and scientists behind it. And often using stories and storytellers with people within our own communities that we trust is going to be a really positive way of doing it. And hopefully people like us in our organization that can help people like you and your communities go out there and talk the stories. And we want to support with the science in the background all we can. So that's our website, 0800 Immune. Within the next day, we're going to get a new look COVID page on this. Within our website, we've got a whole lot of Q's and A's that we're developing. They're really there for myth busting, so that if you hear something weird and wonderful and you want to answer the question, we're trying to have a lot of common Q's and A's on our website so people can go there and check it out, rather than start reiterating and repeating myths that suddenly start getting embedded. We just want them sitting there as a resource for you folks. So that's the resources. Oh, and that's another resource. So I think I'll leave it there. I've talked long enough, and I'll stop share and see if people have got some questions, comments. How can we help? By the way, though, it is a fantastic vaccine, so we've got a really cool product to sell, which is a great starting point. Kia ora.
Kia ora.
So I don't know how you want to manage it at this point. Liz, does anyone want to ask a question or anything in the chat? Or I think there's enough of us that you could just raise your hand and ask a question or a comment or any advice for us. Everything's welcome at this point.
Thank you, Nikki. I mean, it's a really informative, and I think sharing that slide pack will be really helpful for everyone. I don't have any immediate questions, but I'll leave it to the floor. Does anyone, Joel or Max, do you have anything?
Kia ora, Nikki. My name's Joel Pittini. I'm a doc up at Kaitaia Hospital. Thanks very much for the talk, and sorry I was 10 minutes late getting here. You might have covered this at the start of the talk. I was just wondering if there were any other examples of mRNA vaccines that are currently in use in New Zealand.
Yeah, so it was at the start. So there's only 2 licensed mRNA vaccines in the world. That's this Pfizer one and Mrna that the Americans have produced. So mRNA vaccines had not been licensed in human use up till now. So that is one of the early concerns about it. So my point that, yes, they are new. And I don't know if you missed the starting comment that the reason they haven't been is that RNA has been suggested as a vaccine design for many, many years because it's so powerful as a vaccine design. The science breakthrough was the lipid coat that stabilizes the RNA. Once that came through in the last couple of years, then we could proceed with RNA vaccines. So they are new technology, but not entirely new. The RNA technology is not new at all. It was the stabilizing of the lipid coat. Now, what I hope we'll see going on now is they'll now figure out how to make it more stable in the cold chain, because that's really what we'd like to make it easier to deliver. So at this stage, no more licensed RNA vaccines, but a lot in clinical trials.
Yeah, and it was great because I arrived just in time to see that list of ingredients and what they are, because we had a hui here last week at one of the local GP clinics, and the Minister Penehina there was here, and we had a Q&A with some koe and kaumātua, and that was one of the sort of leading questions, is what's in it physically, and have we seen those things before? Are they things that our bodies are exposed to already? And so your slide about that is really helpful in terms of answering that question. Thank you.
Oh, that's great. And we've got some notes underneath it, so we understand, you know, that's the lipid, you know, that's the pH stabilizer, that's the sugar. And a lot of those chemical names look really scary, but most of them are used within our bodies, you know. So I think back to the story point, that when we can translate that scary slide into stories, you know, like we often talk about aluminium adjuvants in other vaccines, not in this one. We say, well, aluminium's in breast milk. So translating them into real life stories is really useful. And this vaccine's a good one to sell because there is actually so little in it.
And did that slide have in it, like, you know, you've got the list of ingredients, did it have some examples of where our bodies are exposed to those things at other times as well?
Yeah, I think mostly in the list, but I mean, most of it is normal products in our body, you know, like the stabilizing product. They're almost all normal products in our body. The major one is that PEG, polyethylene glycol. And in the clinical notes, I've reflected all the different things polyethylene glycol's in. It's very commonly used.
Perfect, thank you.
Max, I think you're on mute.
Sorry, thank you. Kia ora. I'm one of the general surgeons at Whangarei Hospital, Nikki. Thanks so much for your time, and that was really, really useful. I just wondered about mutations. I think that's something that people are just interested in, like how will they, that be detected and addressed with future vaccines or boosters or whatever. Do you?
Yeah, kia ora, good question. So in our bigger slide, our bigger set of slides, which I'll also pass around to Liz, we've got a slide on mutations. So mutations are occurring with that spike protein, and they have occurred, and we're particularly worried about the South African mutation. They don't occur to the same degree as flu vaccines. So we're not going to be in quite the problem we have with flu vaccines, where they mutate regularly all the time. But there have been a couple of difficult mutations where the Pfizer vaccine data to date is suggesting that it's very good against the English mutation, and it's good, maybe slightly blunted against the South African, but it's looking good enough because it's so highly effective. That so far to date, this formulation is okay. If there is a further mutation, then these RNA vaccines, they can very quickly adapt. But of course, that would mean we'd have to boost, be the downside. So there is another conversation going on that these mutations are happening, but at the moment, there haven't been, since the South African and the Brazilian one, any other scary ones. So the worst case scenario is that you reformulate the vaccine and you boost.
And this vaccine just seems so incredible, really. Like, why? I know that we've purchased or right of purchase for the other vaccines. Do you foresee that we might forgo those if this is so successful? Why would we consider the other ones? Yeah.
Well, there's a couple of reasons. One is just the straight logistics. The real pain that we can't just put this in every fridge of every small pharmacy practice. Okay. So logistics are a real bummer. So the other reason for considering other vaccines is we don't know longevity vaccine. So the Janssen vaccine, the vector one, has actually got good data on single dose. So that's quite a huge advantage from a program point of view. And as we watch how long these vaccines last, it might be that mix and matching different sorts of vaccines to give you longer duration of immunity. Those are sort of questions we don't. So I think that, we've got a fantastic vaccine, and I think it's a good call to rocket to the whole population now. But going forward, as we know more about all the vaccines, there might be a different regime in the long term. One will be about pragmatism and one will be about duration of immunity.
Okay, kia ora. I've got one other question. No, people don't mind me jumping in.
Please do. I like the questions.
Do you have a response with the AstraZeneca issue with the thrombosis? Do you have a good response to that when talking to people who, would have ordinarily been a bit hesitant, sorry to use that word, but and then this vaccine's rolled out and now it's pulled, all those kind of things. Is there
So, I think, there's two responses. One is the Pfizer vaccine, that we've got very large clinical data where we've been looking at background rates of incidence like these thrombosis and clots, and there's no clear increase in incidence. The AstraZeneca one was picked up by using the same monitoring system. So one of the reassuring things is this was picked up by monitoring systems. So I think that's reassuring to us that the monitoring systems are working the way they should. Now, it's incredibly rare. It's still really, really rare. So the monitoring systems are picking it up is a sign that that's great. We are seeing these things. And it's unusual blood clotting issues seen probably immune mediated in younger people and more in women. So it wasn't initially picked up in the UK, probably because they rolled it out to elderly people first. Okay. So the Europeans picked it up earlier because they were rolling it out to younger people, because if you remember, they didn't want to use it in the older people first. So that's why the European systems picked it up earlier. The USA wasn't using AstraZeneca and the UK was using it in older. But once it was used in the right age groups, it was picked up. And my next thing I'd say to people is, so that vaccine has been shown to have a signal that suggests that a very rare condition might occur. It's still incredibly rare, but at least we're aware of it and we know it and we can understand it. So great example that the world's safety monitoring is working.
Cool. Kia ora.
Okay, well, I think what you've done is provide us with some wonderful information. Thank you so much for your time today. If nobody else has any questions, I think we can sign it off and share it back to ourselves for sharing with our colleagues.
Kia ora.
So,
I'll send the resources through to Liz, and look, let us know how else we can help folks, because you're the ones out there that are going to be talking the talk. So thanks for your support.
really appreciate your time today. Thank you.
Kia ora
Kia ora,
tata.
Nikki. Thank you.
Hang on one sec, can we just have one?
Sorry, just tēnā koutou. Just one question, time frame of 3 weeks for the second vaccination.
Yeah.
Is that really important?
Yeah, it's a really good question. So actually, I think 3 weeks is really tight. And the reason they did a 3-week gap is because there was an urgency to get people protected when you've got a lot of COVID disease. So that's why the clinical trial chose 3 weeks as the shortest gap immunologically that they thought they could get away with. In a New Zealand context, you don't want to give it less than 3 weeks, because this is what we call a prime boost. And your priming response is still being developed by 3 weeks. So 3 weeks is actually the minimum interval you'd want, but there would not be a problem in delaying it and giving it later.
Nick, please, could I have the first one here and the second one somewhere else? If I
Yeah,
had to go
kia
on?
ora. Absolutely, you could.
Oh, I've just got another question, actually. Sorry, just thinking about diversity of groups that were vaccinated. I mean, I know that huge numbers and things like that. Do you have an idea off the top of your head about the number of, say, Black or Indigenous or minority groups vaccinated as part of the Pfizer trial?
Yeah, so I haven't got it to hand, but if you like, I'll send around the pivotal papers. So I'll send around the pivotal RCT that's got them all listed in it, because it's really good to see. And there's a nice table that lists all the different ethnic groups in it. And I'll also send you around the Israeli paper, which was the original, experiential data. So I think they're both excellent papers to have. Obviously, I believe there were a handful of Pacific people amongst them, but not big numbers, and probably not any Māori at all. But what we know from this clinical trial and from any others is there's no real immunogenicity effectiveness difference by ethnic groups. So we're not expecting it. Now, New Zealand, I believe, is looking at immunogenicity and safety by ethnic group to reassure our population. But I have to say that we've got no evidence in any of our vaccines that there's a difference by ethnic group. So we wouldn't expect there to be a difference, but it's nice that New Zealand's going to check.
And I guess that, you answered the question about multiple comorbidities. Maybe a slight reduction, but still very good. And I guess that's something that would apply particularly to our Māori communities. That would be the divider potentially.
Yeah, I think there's a double message there. So it's a bit like flu vaccine, is that like this is a really effective vaccine, but if your immune system isn't great, then you won't get quite as vigorous a response. So the important, not just of you being vaccinated, but your whānau being vaccinated as well. So somebody who's on oncology drugs or, immunocompromised for some other reason, then the real whānau message is really important. It's like, this is not just about individual protection. And I like that message. I think that's a really strong message. It's one we try to use with MMR as well. That, some people, you can all have this vaccine, but it might not work so well for people whose immune system is really not working very well. So make sure the rest of your whānau have had it.
Hey, Nikki, is there any data or information that you have about, because I think the two actually most important things raised in your talk that we need to focus on is about community confidence and reaching those who miss out. Like we're all convinced that this works, but part of the challenge is convincing the community and getting them to buy in and eat the cake. So again, reflecting on minority groups or different ethnicities rather than European ethnicities, is there any particular data or resource you have around how they've done it in other places, and have they had success rates in terms of getting access and equity? Yeah,
there's masses of answers to that. None of them are clear. So each community is a little different. And I think without diving into the data too much, the answer is really, it's about trusted people communicating, however you pitch it and however it looks. So firstly, for the community to understand and feel heard, there's a lot of data out there that communities feel too scared to speak up and not heard. So, you know, people being allowed to ask what they see as the dumb questions, or being allowed to say, I'm really scared. The other thing I think I haven't included, but I should have, is a lot of people are needle phobic, and we often miss that. We picked that up with the Pacific community early on. A lot of people just missing the fact that actually, quite frankly, they don't want to come for a needle. So acknowledging a lot of people's genuine fears is really important. And then it's communicating it within the voice and the sounds within your own community. So that's essentially what it all says. And then the health services being responsive to the community and not creating barriers. So, you know, so that's why I think hesitancy is the wrong word. You know, there's all these barriers we create. So there's a huge amount of different pieces of information, and none of them directly, I can't give you one that says, here's the single recipe that works. But just if you work on trust, allowing people to be heard, reflecting all their questions are valid, and walking alongside them. I've never found, you know, getting angry or telling people they're stupid, or just repeating vaccines are safe, vaccines are safe, vaccines are safe, is completely the wrong way to go, you know. So that's my starting point. I'm not sure how useful that is for you.
No, that's, I mean, I guess I asked that question because I've got Hepa Stevens here, who's our kaitaka wāinga for the hospital, and Hep and I have been talking a lot about organising hui around town, inviting community and having Q&A sessions. I guess what I didn't want to do is do that, and it's already been proven that that's a waste of time and doesn't work. But
the communication session's great.
Sounds like that is a tool that's been used and has been shown to be effective. So we'll go ahead and keep doing
And
it.
we've got different pieces of resources, so keep talking to us. We're also working with Jess Berenson Shaw from the Ideas Shop to try and help with a communication manual that we can pass on to you guys, and then, you know, to reflect those sort of messaging, like that. Those 2 slides I put in there were from Jess Berenson Shaw, to reflect those sort of messaging when you go out with your communications. I think might be quite helpful. So keep talking to us. I just introduced Denise, who's online at the moment. Denise, maybe you want to just briefly introduce yourself, and Denise would be the conduit to IMAC to help you guys with some of these. So Denise, do you want to make a few comments and just introduce yourself?
Kia whānau, he mihi iti tēnei kia kaukata katoa, he ria hau no Ngāti Awa, ngai tūhoi Ngāti Kahununu ki te Wairua, ko tāka mahi ka wanganui I te whānau IMAC, ko au te kaiwhakahaere Māori mō te katoa o Aotearoa I pāna ki ngā kaupapa Māori. So, kia ora everybody, I hail from Whakatane. I moved up to Auckland about a month and a half ago, and basically I come with a background in Māori engagement and Māori engagement strategies for rural and urban cities. So I come with a strong rural background and engaging Māori communities and Māori whānau, and with a focus at grassroots around engaging effectively with whānau, and then working with providers to be able to provide the effective and correct communication to involve whānau hapu and iwi in terms of not only finding out information on what they want to hear, but also on how they want us to share the kōrero or to share those pūrākau with them. So I work at IMAC as the Māori Engagement Advisor for IMAC, which sits at a national role across Aotearoa, and really one of the biggest things in my role is working with whānau hapu and iwi and also providers to make sure that the kupu, the kōrero, everything has a te ao Māori focus on it, so that we can best communicate to our whānau hapu and iwi in an effective way in which they see it. So that's me, whānau. Kia ora.
Kia ora, Denise.
Kia ora, Denise. That is so exciting because Chanel is with us today. She's on our communications team. We were just starting to prepare some community hui for motivational conversation training, hopefully in partnership with IMAC. So it seems to me that we will be able to partner up with someone of your skill set and experience, because we think that if we are able to empower all of our healthcare workers, Māori and non-Māori, then we increase greatly the number of positive conversations and that we can have out there.
Definitely, and I think one of the biggest things that I've gotten from the Māori communities, Māori providers, is we actually want words that we use all the time, and we want words that are very familiar to us as Māori, and metaphors that are familiar to us as Māori. So that's something that I've been working alongside our education team to help awhi them with, particularly in our rural communities, so that we're giving kōrero that is in a way that our whānau, hapu, iwi, and even Māori collective understand, but also too in words that are simple and metaphors that are easy to understand for our whānau. So tēnā koutou.
Actually, kia ora tātou, everyone. Ko Michelle Bristow, tōku ingoa. I'm the Māori comms response lead with the NRHCC. So at the moment, sorry I was a bit late, I'm at Manurewa Marae, been here this morning. So the vaccination started today. And if you want to, I can show you what's been happening. But actually the engagement with our communities is incredibly important. You're quite right, it'd be lovely to meet you, Denise. Because one of our strategies has been, and this is probably the start of it, is that empowering our kaumahi in the field to be able to have those conversations, to be able from the receptionist through to the, to the clinician to say, look, if anyone talks to them about it, they can at least have a basic conversation about the vaccine, and that they are confident in what they're saying. But the second part of it actually kind of covers everyone, is that we're looking at doing roadshows. That's our plan, is to get out from behind the computer, out from behind our posters, and to talk to our whānau and our community. So the Q&A you were talking about wanting earlier is something that we're looking at doing and planning in conjunction with iwi around, where our remit is, which is the northern region, actually. So, but, like today here at Manurewa, it was an interesting exercise, listening to people. And I just had someone before I came online sit down next to me, and she walked to the door of the wharenui, which is where they're holding their vaccine. So again, it's a familiar space. It's somewhere that they have been to a lot. But she walked backwards and forwards about 3 times. And the reason she did it, she says, I'm scared to go in. And I said, why? And she says, I'm not keen on needles, and she says, what happens? Does it? So it's actually probably more around people's phobias sometimes about, and actually COVID has been, the word COVID has become something of almost, has something a lot of people are fearing. So even the cure is a fear that they have to overcome. And I think that that's something that is saying that, our messaging does need to be something that they can connect with, but also that they are saying that we are here to help. This is something that's good. This is something that's going to get us through what the world is going through at the moment. So yeah, we'll keep you all informed. So thanks everybody for being here.
Kia ora, Michelle. I just want to say I'm a Manarewa kid. I grew up in Manarewa, so heart and soul, I'm a county's kid.
Okay, well, I think unless I'll do one more round, if anyone has any questions, I'd like to really acknowledge everyone's time today. Thank you very much, Nikki. Oh, there we go. We're out in the field.
Fabulous.
Thank you, Maxine, for joining us at home. Joel, Hepa, thank you, and everybody else. So we'll sign off now. Kia ora koutou.
Kia ora. Keep in touch.
Thanks.
Thanks.
Anō.
Thank you.
Kia ora.
Ka kite whānau.